Photo of chickenpox rash on the back of a person's shoulders. Figure 17. 1. Varicella, or chickenpox, is caused by the highly contagious varicella-zoster virus. The characteristic rash seen here is partly a result of inflammation associated with the body's immune response to the virus. Inflammation is a response mechanism of innate immunity that helps the body fight off a wide range of infections. Credit John Noble/CDC, Public Domain, Chapter Outline 17. 1. Physical defenses 17. 2. Chemical defenses 17. 3. Cellular defenses 17. 4. Pathogen recognition and phagocytosis 17. 5. Inflammation and fever despite relatively constant exposure to pathogenic microbes in the environment. Humans do not generally suffer from constant infection or disease. Under most circumstances, the body is able to defend itself from the threat of infection, thanks to a complex immune system designed to repel, kill, and expel disease-causing invaders. Immunity as a whole can be described as two interrelated parts: nonspecific innate immunity, which is the subject of this chapter, and specific adaptive host defenses, which are discussed in the next chapter. The nonspecific innate immune response provides a first line of defense that can often prevent infections from gaining a solid foothold in the body. These defenses are described as nonspecific because they do not target any specific pathogen. Rather, they defend against a wide range of potential pathogens. They are called innate because they are built-in mechanisms of the human organism. Unlike the specific adaptive defenses, they are not acquired over time, and they have no memory. They do not improve after repeated exposures to specific pathogens. Broadly speaking, nonspecific innate defenses provide an immediate, or very rapid, response against potential pathogens. However, these responses are neither perfect nor impenetrable. They can be circumvented by pathogens on occasion, and sometimes they can even cause damage to the body, contributing to the signs and symptoms of infection. Figure 17.1. Learning objectives. By the end of this section, you will be able to describe the various physical barriers and mechanical defenses that protect the human body against infection and disease. Describe the role of microbiota as a first-line defense against infection and disease. Clinical focus. Part 1. Angela, a 25-year-old female patient in the emergency department, is having some trouble communicating verbally because of shortness of breath. A nurse observes constriction and swelling of the airway and labored breathing. The nurse asks Angela if she has a history of asthma or allergies. Angela shakes her head no, but there is fear in her eyes. With some difficulty, she explains that her father died suddenly at age 27, when she was just a little girl, of a similar respiratory attack. The underlying cause had never been identified. What are some possible causes of constriction and swelling of the airway? What causes swelling of body tissues in general? Jump to the next clinical focus box. Nonspecific innate immunity can be characterized as a multifaceted system of defenses that targets invading pathogens in a nonspecific manner. In this chapter, we have divided the numerous defenses that make up this system into three categories: physical defenses, chemical defenses, and cellular defenses. However, it is important to keep in mind that these defenses do not function independently, and the categories often overlap. Table 17. 1 provides an overview of the nonspecific defenses discussed in this chapter. Overview of nonspecific innate immune defenses, physical barriers, mechanical defenses, microbiome, chemical defenses, chemicals and enzymes, in body fluids, antimicrobial peptides, plasma protein mediators, cytokines, inflammation-eliciting mediators, cellular defenses, granulocytes, agranulocytes. Table 17. 1. Physical defenses provide the body's most basic form of nonspecific defense. They include physical barriers to microbes, such as the skin and mucous membranes, as well as mechanical defenses that physically remove microbes and debris from areas of the body where they might cause harm or infection. In addition, the microbiome provides a measure of physical protection against disease, as microbes of the normal microbiota compete with pathogens for nutrients and cellular binding sites necessary to cause infection. Physical barriers play an important role in preventing microbes from reaching tissues that are susceptible to infection. At the cellular level, barriers consist of cells that are tightly joined to prevent invaders from crossing through to deeper tissue. For example, the endothelial cells that line blood vessels have very tight cell-to-cell junctions, blocking microbes from gaining access to the bloodstream. Cell junctions are generally composed of cell membrane proteins that may connect with the extracellular matrix or with complementary proteins from neighboring cells. Tissues in various parts of the body have different types of cell junctions. These include tight junctions, desmosomes, and gap junctions, as illustrated in Figure 17. 2. Invading microorganisms may attempt to break down these substances chemically, using enzymes such as proteases that can cause structural damage to create a point of entry for pathogens. Tight junctions, two membranes connected with many spot welds in multiple lines. Desmosomes, two membranes with long strands weaving them together. Gap junctions, two membranes with a few spot welds, each of which has a pore in the center. Figure 17. 2. There are multiple types of cell junctions in human tissue, three of which are shown here. Tight junctions rivet two adjacent cells together, preventing or limiting material exchange through the spaces between them. Desmosomes have intermediate fibers that act like shoelaces, tying two cells together, allowing small materials to pass through the resulting spaces. Gap junctions are channels between two cells that permit their communication via signals. Credit, modification of work by Mariana Ruiz Villarreal. The skin barrier, one of the body's most important physical barriers, is the skin barrier, which is composed of three layers of closely packed cells. The thin upper layer is called the epidermis. A second, thicker layer called the dermis contains hair follicles, sweat glands, nerves, and blood vessels. A layer of fatty tissue called the hypodermis lies beneath the dermis and contains blood and lymph vessels, Figure 17. 3. A diagram of a section of skin. The bottom layer is the hypodermis and is mostly made up of large circular cells, fatty tissue. The next layer up, and the thickest layer is the dermis. At the bottom of the dermis are blood vessels, lymph vessels, and nerves, all of which run throughout the dermis. Sweat glands are coiled tubes that lead to the surface. Hair follicles are thick vase-shaped structures containing a hair. An oil gland is attached to the hair follicle. The top layer is the epidermis and is made of many layers of flat cells. Figure 17. 3. Human skin has three layers, the epidermis, the dermis, and the hypodermis, which provide a thick barrier between microbes outside the body and deeper tissues. Dead skin cells on the surface of the epidermis are continually shed, taking with them microbes on the skin's surface. Credit, modification of work by National Institutes of Health. The topmost layer of skin, the epidermis, consists of cells that are packed with keratin. These dead cells remain as a tightly connected, dense layer of protein-filled cell husks on the surface of the skin. The keratin makes the skin's surface mechanically tough and resistant to degradation by bacterial enzymes. Fatty acids on the skin's surface create a dry, salty, and acidic environment that inhibits the growth of some microbes and is highly resistant to breakdown by bacterial enzymes. In addition, the dead cells of the epidermis are frequently shed, along with any microbes that may be clinging to them. Shed skin cells are continually replaced with new cells from below, providing a new barrier that will soon be shed in the same way. Infections can occur when the skin barrier is compromised or broken. A wound can serve as a point of entry for opportunistic pathogens, which can infect the skin tissue surrounding the wound and possibly spread to deeper tissues. Case in point, every rose has its thorn. Mike, a gardener from Southern California, recently noticed a small red bump on his left forearm. Initially, he did not think much of it, but soon it grew larger and then ulcerated, opened up, becoming a painful lesion that extended across a large part of his forearm, Figure 17. 4. He went to an urgent care facility, where a physician asked about his occupation. When he said he was a landscaper, the physician immediately suspected a case of sporotrichosis, a type of fungal infection known as rose gardener's disease, because it often afflicts landscapers and gardening enthusiasts. Under most conditions, fungi cannot produce skin infections in healthy individuals. Fungi grow filaments known as hyphae, which are not particularly invasive and can be easily kept at bay by the physical barriers of the skin and mucous membranes. However, small wounds in the skin, such as those caused by thorns, can provide an opening for opportunistic pathogens like Sporothrix shankii, a soil-dwelling fungus, and the causative agent of Rose Gardner's disease. Once it breaches the skin barrier, S. shankii can infect the skin and underlying tissues, producing ulcerated lesions like mites. Compounding matters, other pathogens may enter the infected tissue, causing secondary bacterial infections. Luckily, Rose Gardner's disease is treatable. Mike's physician wrote him a prescription for some antifungal drugs, as well as a course of antibiotics, to combat secondary bacterial infections. His lesions eventually healed, and Mike returned to work with a new appreciation for gloves and protective clothing. An arm with festering, bleeding scratches, and a picture of a rose plant. Figure 17. 4. Rose Gardner's disease can occur when the fungus Sporothrix shankii breaches the skin through small cuts, such as might be inflicted by thorns. Credit left. Modification of work by Alyssa Self. Credit right. Modification of work by Centers for Disease Control and Prevention. Mucous membranes. The mucous membranes lining the nose, mouth, lungs, and urinary and digestive tracts provide another nonspecific barrier against potential pathogens. Mucous membranes consist of a layer of epithelial cells bound by tight junctions. The epithelial cells secrete a moist, sticky substance called mucus, which covers and protects the more fragile cell layers beneath it and traps debris and particulate matter, including microbes. Mucus secretions also contain antimicrobial peptides. In many regions of the body, mechanical actions serve to flush mucus, along with trapped or dead microbes, out of the body or away from potential sites of infection. For example, in the respiratory system, inhalation can bring microbes, dust, mold spores, and other small airborne debris into the body. This debris becomes trapped in the mucus lining the respiratory tract, a layer known as the mucociliary blanket. The epithelial cells lining the upper parts of the respiratory tract are called ciliated epithelial cells because they have hair-like appendages known as cilia. Movement of the cilia propels debris-laden mucus out and away from the lungs. The expelled mucus is then swallowed and destroyed in the stomach, or coughed up, or sneezed out. Figure 17.5. This system of removal is often called the mucociliary escalator, a spongy-looking surface with tufts of long hairs. Each hair is about 5 microM long, each tuft is about 10 microM in diameter. Figure 17. 5. This scanning electron micrograph shows ciliated and non-ciliated epithelial cells from the human trachea. The mucociliary escalator pushes mucus away from the lungs, along with any debris or microorganisms that may be trapped in the sticky mucus, and the mucus moves up to the esophagus, where it can be removed by swallowing. The mucociliary escalator is such an effective barrier to microbes that the lungs, the lowermost and most sensitive portion of the respiratory tract, were long considered to be a sterile environment in healthy individuals. Only recently has research suggested that healthy lungs may have a small normal microbiota. Disruption of the mucociliary escalator by the damaging effects of smoking or diseases, such as cystic fibrosis, can lead to increased colonization of bacteria in the lower respiratory tract and frequent infections, which highlights the importance of this physical barrier to host defenses. Like the respiratory tract, the digestive tract is a portal of entry through which microbes enter the body, and the mucous membranes lining the digestive tract provide a nonspecific physical barrier against ingested microbes. The intestinal tract is lined with epithelial cells, interspersed with mucus-secreting goblet cells. Figure 17. 6. This mucus mixes with material received from the stomach, trapping foodborne microbes and debris. The mechanical action of peristalsis, a series of muscular contractions in the digestive tract, moves the slewed mucus and other material through the intestines, rectum, and anus, excreting the material in feces. Figure A is a diagram of a single goblet cell. Cell is tall and slightly hourglass-shaped. The bottom of the cell is filled with a nucleus. The top shows the Golgi apparatus folds of membranes, rough endoplasmic reticulum folds of membranes with dots, secretory vesicles containing mucin large bubbles, and microvilli finger-like projections at the top. Figure B is a micrograph of two goblet cells within a row of epithelial cells. The epithelial cells are rectangular with a large nucleus visible. The goblet cells are thinner and have a clear uncolored top. Figure 17. 6 goblet cells produce and secrete mucus. The arrows in this micrograph point to the mucus-secreting goblet cells, magnification 1600, in the intestinal epithelium. Credit micrograph. Micrograph provided by the Regents of University of Michigan Medical School, copyright 2012. Endothelia, the epithelial cells lining the urogenital tract, blood vessels, lymphatic vessels, and certain other tissues are known as endothelia. These tightly packed cells provide a particularly effective frontline barrier against invaders. The endothelia of the blood-brain barrier, for example, protect the central nervous system, CNS, which consists of the brain and the spinal cord. The CNS is one of the most sensitive and important areas of the body, as microbial infection of the CNS can quickly lead to serious and often fatal inflammation. The cell junctions in the blood vessels traveling through the CNS are some of the tightest and toughest in the body, preventing any transient microbes in the bloodstream from entering the CNS. This keeps the cerebrospinal fluid that surrounds and bathes the brain and spinal cord sterile under normal conditions. Check your understanding. Describe how the mucociliary escalator functions. Name 2 places you would find endothelia. Mechanical defenses. In addition to physical barriers that keep microbes out, the body has a number of mechanical defenses that physically remove pathogens from the body, preventing them from taking up residence. We have already discussed several examples of mechanical defenses, including the shedding of skin cells, the expulsion of mucus via the mucociliary escalator, and the excretion of feces through intestinal peristalsis. Other important examples of mechanical defenses include the flushing action of urine and tears, which both serve to carry microbes away from the body. The flushing action of urine is largely responsible for keeping the urinary tract, which includes the kidneys, ureters, and urinary bladder, free of transient microorganisms that may include pathogens. Urine passing out of the body washes out transient microorganisms, preventing them from taking up residence. The eyes also have physical barriers and mechanical mechanisms for preventing infections. The eyelashes and eyelids prevent dust and airborne microorganisms from reaching the surface of the eye. Any microbes or debris that make it past these physical barriers may be flushed out by the mechanical action of blinking, which bathes the eye in tears, washing debris away. Figure 17. 7. A diagram of a person. An arrow from the eye points to a larger image that shows eyelashes, the eyelid and tear ducts. An arrow from the abdominal region shows a larger kidney or ureter. An arrow from the groin region shows a larger bladder, including ureter, pelvic floor muscles, and urethra. Figure 17. 7 tears flush microbes away from the surface of the eye. Urine washes microbes out of the urinary tract as it passes through. As a result, the urinary system is normally sterile. Check your understanding. Name 2 mechanical defenses that protect the eyes. Microbiome in various regions of the body, resident microbiota, serve as an important first-line defense against invading pathogens. Through their occupation of cellular binding sites and competition for available nutrients, the resident microbiota prevent the critical early steps of pathogen attachment and proliferation required for the establishment of an infection. For example, in the vagina, members of the resident microbiota compete with opportunistic pathogens like the yeast Candida. This competition prevents infections by limiting the availability of nutrients, thus inhibiting the growth of Candida, keeping its population in check. Similar competitions occur between the microbiota and potential pathogens on the skin, in the upper respiratory tract, and in the gastrointestinal tract. As will be discussed later in this chapter, the resident microbiota also contribute to the chemical defenses of the innate nonspecific host defenses. The importance of the normal microbiota in host defenses is highlighted by the increased susceptibility to infectious diseases when the microbiota is disrupted or eliminated. Treatment with antibiotics can significantly deplete the normal microbiota of the gastrointestinal tract, providing an advantage for pathogenic bacteria to colonize and cause diarrheal infection. In the case of diarrhea caused by Clostridioides difficile, the infection can be severe and potentially lethal. One strategy for treating C. difficile infections is fecal transplantation, which involves the transfer of fecal material from a donor screened for potential pathogens into the intestines of the recipient patient as a method of restoring the normal microbiota and combating C. difficile infections. Table 17. 2 provides a summary of the physical defenses discussed in this section. Physical defenses of nonspecific innate immunity defense examples function cellular barriers skin, mucous membranes, endothelial cells deny entry to pathogens mechanical defenses shedding of skin cells, mucociliary sweeping. Peristalsis, flushing action of urine and tears remove pathogens from potential sites of infection, microbiome resident bacteria of the skin, upper respiratory tract, gastrointestinal tract, and genitourinary tract compete with pathogens for cellular binding sites and nutrients. Table 17. 2. Check your understanding list. 2 ways resident microbiota defend against pathogens. Learning objectives. By the end of this section, you will be able to describe how enzymes in body fluids provide protection against infection or disease list, and describe the function of antimicrobial peptides, complement components, cytokines, and acute phase proteins. Describe similarities and differences among classic, alternate, and lectin complement pathways, in addition to physical defenses. The innate nonspecific immune system uses a number of chemical mediators that inhibit microbial invaders. The term chemical mediators encompasses a wide array of substances found in various body fluids and tissues throughout the body. Chemical mediators may work alone or in conjunction with each other to inhibit microbial colonization and infection. Some chemical mediators are endogenously produced, meaning they are produced by human body cells. Others are produced exogenously, meaning that they are produced by certain microbes that are part of the microbiome. Some mediators are produced continually, bathing the area in the antimicrobial substance. Others are produced or activated primarily in response to some stimulus, such as the presence of microbes. Chemical and enzymatic mediators found in body fluids produced by the skin include examples of both endogenous and exogenous mediators. Sebaceous glands in the dermis secrete an oil called sebum that is released onto the skin surface through hair follicles. This sebum is an endogenous mediator, providing an additional layer of defense by helping seal off the pore of the hair follicle, preventing bacteria on the skin's surface from invading sweat glands and surrounding tissue. Figure 17. 8. Certain members of the microbiome, such as the bacterium Cutibacterium acnes and the fungus Malassezia, among others, can use lipase enzymes to degrade sebum, using it as a food source. This produces oleic acid, which creates a mildly acidic environment on the surface of the skin that is inhospitable to many pathogenic microbes. Oleic acid is an example of an exogenously produced mediator because it is produced by resident microbes and not directly by body cells. A micrograph and diagram both show a large hair follicle, a vase-shaped pocket, with a hair projecting out past the epidermis. On the side of the hair follicle is the sebaceous gland, which is a lumpy structure. Figure 17. 8 sebaceous glands secrete sebum, a chemical mediator that lubricates and protects the skin from invading microbes. Sebum is also a food source for resident microbes that produce oleic acid, an exogenously produced mediator. Credit micrograph. Micrograph provided by the Regents of University of Michigan Medical School, copyright 2012. Environmental factors that affect the microbiota of the skin can have a direct impact on the production of chemical mediators. Low humidity or decreased sebum production, for example, could make the skin less habitable for microbes that produce oleic acid, thus making the skin more susceptible to pathogens normally inhibited by the skin's low pH. Many skin moisturizers are formulated to counter such effects by restoring moisture and essential oils to the skin. The digestive tract also produces a large number of chemical mediators that inhibit or kill microbes. In the oral cavity, saliva contains mediators such as lactoperoxidase enzymes, and mucus secreted by the esophagus contains the antibacterial enzyme lysozyme. In the stomach, highly acidic gastric fluid kills most microbes. In the lower digestive tract, The intestines have pancreatic and intestinal enzymes, antibacterial peptides, cryptins, bile produced from the liver, and specialized paineth cells that produce lysozyme. Together, these mediators are able to eliminate most pathogens that manage to survive the acidic environment of the stomach. In the urinary tract, urine flushes microbes out of the body during urination. Furthermore, the slight acidity of urine, the average pH is about 6, inhibits the growth of many microbes and potential pathogens in the urinary tract. The female reproductive system employs lactate, an exogenously produced chemical mediator, to inhibit microbial growth. The cells and tissue layers composing the vagina produce glycogen, a branched and more complex polymer of glucose. Lactobacilli in the area ferment glycogen to produce lactate, lowering the pH in the vagina and inhibiting transient microbiota, opportunistic pathogens like Candida, a yeast associated with vaginal infections, and other pathogens responsible for sexually transmitted diseases. In the eyes, tears contain the chemical mediators lysozyme and lactoferrin, both of which are capable of eliminating microbes that have found their way to the surface of the eyes. Lysozyme cleaves the bond between NAG and NOM in peptidoglycan, a component of the cell wall in bacteria. It is more effective against gram-positive bacteria, which lack the protective outer membrane associated with gram-negative bacteria. Lactoferrin inhibits microbial growth by chemically binding and sequestering iron. This effectually starves many microbes that require iron for growth. In the ears, cerumen earwax exhibits antimicrobial properties due to the presence of fatty acids, which lower the pH to between 3 and 5. The respiratory tract uses various chemical mediators in the nasal passages, trachea, and lungs. The mucus produced in the nasal passages contains a mix of antimicrobial molecules similar to those found in tears and saliva, e.g., lysozyme, lactoferrin, lactoperoxidase. Secretions in the trachea and lungs also contain lysozyme and lactoferrin, as well as a diverse group of additional chemical mediators, such as the lipoprotein complex called surfactant, which has antibacterial properties. Check your understanding, explain the difference between endogenous and exogenous mediators, describe how pH affects antimicrobial defenses, antimicrobial peptides, the antimicrobial peptides, AMPs, are a special class of nonspecific cell-derived mediators with broad-spectrum antimicrobial properties. Some AMPs are produced routinely by the body, whereas others are primarily produced, or produced in greater quantities, in response to the presence of an invading pathogen. Research has begun exploring how AMPs can be used in the diagnosis and treatment of disease. AMPs may induce cell damage in microorganisms in a variety of ways, including by inflicting damage to membranes, destroying DNA and RNA, or interfering with cell wall synthesis. Depending on the specific antimicrobial mechanism, a particular AMP may inhibit only certain groups of microbes, e.g., gram-positive or gram-negative bacteria, or it may be more broadly effective against bacteria, fungi, protozoa, and viruses. Many AMPs are found on the skin, but they can also be found in other regions of the body. A family of AMPs called defensins can be produced by epithelial cells throughout the body as well as by cellular defenses such as macrophages and neutrophils. See cellular defenses. Defensins may be secreted or act inside host cells. They combat microorganisms by damaging their plasma membranes. AMPs called bacteriocins are produced exogenously by certain members of the resident microbiota within the gastrointestinal tract. The genes coding for these types of AMPs are often carried on plasmids and can be passed between different species within the resident microbiota through lateral or horizontal gene transfer. There are numerous other AMPs throughout the body. The characteristics of a few of the more significant AMPs are summarized in Table 17. 3. Characteristics of selected antimicrobial peptides. AMPs. AMPs secreted by body site pathogens, inhibited mode of action, bacteriocins, resident microbiota gastrointestinal tract bacteria, disrupt membrane cathelicidin epithelial cells. Macrophages and other cell types skin bacteria and fungi disrupts membrane defensins epithelial cells, macrophages, neutrophils throughout the body fungi, bacteria. And many viruses disrupt membrane dermcidins, sweat glands, skin bacteria, and fungi, disrupts membrane integrity, and ion channels histidines, salivary glands, oral cavity fungi, disrupt intracellular function. Table 17. 3. Check your understanding. Why are antimicrobial peptides, AMPs, considered nonspecific defenses? Plasma protein mediators. Many nonspecific innate immune factors are found in plasma, the fluid portion of blood. Plasma contains electrolytes, sugars, lipids, and proteins, each of which helps to maintain homeostasis, i.e., stable internal body functioning, and contains the proteins involved in the clotting of blood. Additional proteins found in blood plasma, such as acute phase proteins, complement proteins, and cytokines, are involved in the nonspecific innate immune response. Microconnections, plasma versus serum. There are two terms for the fluid portion of blood: plasma and serum. How do they differ if they are both fluid and lack cells? The fluid portion of blood left over after coagulation, blood cell clotting, has taken place is serum. Although molecules such as many vitamins, electrolytes, certain sugars, complement proteins, and antibodies are still present in serum, clotting factors are largely depleted. Plasma, conversely, still contains all the clotting elements. To obtain plasma from blood, an anticoagulant must be used to prevent clotting. Examples of anticoagulants include heparin and ethylene diamine tetracetic acid, EDTA. Because clotting is inhibited, once obtained, the sample must be gently spun down in a centrifuge. The heavier, denser blood cells form a pellet at the bottom of a centrifuge tube, while the fluid plasma portion, which is lighter and less dense, remains above the cell pellet. Acute phase proteins. The acute phase proteins are another class of antimicrobial mediators. Acute phase proteins are primarily produced in the liver and secreted into the blood in response to inflammatory molecules from the immune system. Examples of acute phase proteins include C-reactive protein, serum amyloid A, ferritin, transferrin, fibrinogen, and mannose-binding lectin. Each of these proteins has a different chemical structure and inhibits or destroys microbes in some way. Table 17. 4. Some acute phase proteins and their functions. C-reactive protein coats bacteria opsonization, preparing them for ingestion by phagocyte serum, amyloid of ferritin bind and sequester iron, thereby inhibiting the growth of pathogens. Transferrin fibrinogen, involved in formation of blood clots that trap bacterial pathogens. Mannose-binding lectin activates complement cascade. Table 17. For the complement system is a group of plasma protein mediators that can act as an innate nonspecific defense, while also serving to connect innate and adaptive immunity, discussed in the next chapter. The complement system is composed of more than 30 proteins, including C1 through C9, that normally circulate as precursor proteins in blood. These precursor proteins become activated when stimulated or triggered by a variety of factors, including the presence of microorganisms. Complement proteins are considered part of innate nonspecific immunity because they are always present in the blood and tissue fluids, allowing them to be activated quickly. Also, when activated through the alternative pathway, described later in this section, complement proteins target pathogens in a nonspecific manner. The process by which circulating complement precursors become functional is called complement activation. This process is a cascade that can be triggered by one of three different mechanisms, known as the alternative, classical, and lectin pathways. The alternative pathway is initiated by the spontaneous activation of the complement protein C3. The hydrolysis of C3 produces two products, C3a and C3b. When no invader microbes are present, C3b is very quickly degraded in a hydrolysis reaction using the water in the blood. However, if invading microbes are present, C3b attaches to the surface of these microbes. Once attached, C3b will recruit other complement proteins in a cascade. Figure 17. 9. The classical pathway provides a more efficient mechanism of activating the complement cascade, but it depends upon the production of antibodies by the specific adaptive immune defenses. To initiate the classical pathway, a specific antibody must first bind to the pathogen to form an antibody-antigen complex. This activates the first protein in the complement cascade, the C1 complex. The C1 complex is a multi-part protein complex, and each component participates in the full activation of the overall complex. Following recruitment and activation of the C1 complex, the remaining classical pathway complement proteins are recruited and activated in a cascading sequence, figure 17. 9. The lectin activation pathway is similar to the classical pathway, but it is triggered by the binding of mannose-binding lectin, an acute phase protein, to carbohydrates on the microbial surface. Like other acute phase proteins, lectins are produced by liver cells and are commonly upregulated in response to inflammatory signals received by the body during an infection, figure 17. 9. A diagram outlining the three complement pathways. At the top is the invading pathogen. Two antibodies bind to an antigen on the surface of the pathogen. C1 binds to the antigen-antibody complex. This is labeled the classic pathway. C1 causes C2 and C4 to be cut into two pieces. Parts of C2 and C4 bind together to form C3 convertase. The alternate pathway also leads to C3 convertase, but does so directly. C3 convertase then cuts C3 in two, and one of these binds to C3 convertase. The resulting enzyme is called C5 convertase. C5 convertase lyses C5 into two pieces. One of the C5 pieces joins other complement proteins, C6, C7, C8, and C9, to create a pore through the membrane of the invading cell. This pore kills the cell. Endogenous proteins on the host cell protect the host membrane from the complement proteins. Figure 17. 9. The three complement activation pathways have different triggers, as shown here, but all three result in the activation of the complement protein C3, which produces C3a and C3b. The latter binds to the surface of the target cell and then works with other complement proteins to cleave C5 into C5a and C5b. C5b also binds to the cell surface and then recruits C6 through C9. These molecules form a ring structure called the membrane attack complex (MAC), which punches through the cell membrane of the invading pathogen, causing it to swell and burst. Although each complement activation pathway is initiated in a different way, they all provide the same protective outcomes: opsonization, inflammation, chemotaxis, and cytolysis. The term opsonization refers to the coating of a pathogen by a chemical substance, called an opsonin, that allows phagocytic cells to recognize, engulf, and destroy it more easily. Opsonins from the complement cascade include C1q, C3b, and C4b. Additional important opsonins include mannose-binding proteins and antibodies. The complement fragments C3a and C5a are well-characterized anaphylatoxins with potent proinflammatory functions. Anaphylatoxins activate mast cells, causing degranulation and the release of inflammatory chemical signals, including mediators that cause vasodilation and increased vascular permeability. C5a is also one of the most potent chemoattractants for neutrophils and other white blood cells, cellular defenses that will be discussed in the next section. The complement proteins C6, C7, C8, and C9 assemble into a membrane attack complex (MAC), which allows C9 to polymerize into pores in the membranes of gram-negative bacteria. These pores allow water, ions, and other molecules to move freely in and out of the targeted cells, eventually leading to cell lysis and death of the pathogen, figure 17. 9. However, the MAC is only effective against gram-negative bacteria. It cannot penetrate the thick layer of peptidoglycan associated with cell walls of gram-positive bacteria. Since the MAC does not pose a lethal threat to gram-positive bacterial pathogens, complement-mediated opsonization is more important for their clearance. Cytokines are soluble proteins that act as communication signals between cells. In a nonspecific innate immune response, various cytokines may be released to stimulate production of chemical mediators or other cell functions, such as cell proliferation, cell differentiation, inhibition of cell division, apoptosis, and chemotaxis. When a cytokine binds to its target receptor, the effect can vary widely depending on the type of cytokine and the type of cell or receptor to which it has bound. The function of a particular cytokine can be described as autocrine, paracrine, or endocrine, figure 17. 10. In autocrine function, the same cell that releases the cytokine is the recipient of the signal. In other words, autocrine function is a form of self-stimulation by a cell. In contrast, paracrine function involves the release of cytokines from one cell to other nearby cells, stimulating some response from the recipient cells. Last, endocrine function occurs when cells release cytokines into the bloodstream to be carried to target cells much farther away. Cytokines are molecular messengers. In autocrine signaling, the same cell secretes and receives cytokine signals. The diagram shows a single cell releasing molecules and having the molecules bind to receptors on its surface. In paracrine signaling, cytokine signals are secreted to a nearby cell. The diagram shows a cell labeled secreting cell secreting cytokines. A nearby cell has receptors for the molecules. In endocrine signaling, cytokine signals are secreted to the circulatory system and travel to distant cells. The diagram shows the secreting cell secreting cytokines. The cytokines then travel through a blood vessel and bind to receptors on a distant cell. Figure 17. 10 autocrine, paracrine, and endocrine actions describe which cells are targeted by cytokines and how far the cytokines must travel to bind to their intended target cells, receptors. 3 important classes of cytokines are the interleukins, chemokines, and interferons. The interleukins were originally thought to be produced only by leukocytes, white blood cells, and to only stimulate leukocytes, thus the reasons for their name. Although interleukins are involved in modulating almost every function of the immune system, their role in the body is not restricted to immunity. Interleukins are also produced by and stimulate a variety of cells unrelated to immune defenses. The chemokines are chemotactic factors that recruit leukocytes to sites of infection, tissue damage, and inflammation. In contrast to more general chemotactic factors, like complement factor C5a, chemokines are very specific in the subsets of leukocytes they recruit. Interferons are a diverse group of immune signaling molecules and are especially important in our defense against viruses. Type I interferons, interferon alpha and interferon beta, are produced and released by cells infected with virus. These interferons stimulate nearby cells to stop production of mRNA, destroy RNA already produced, and reduce protein synthesis. These cellular changes inhibit viral replication and production of mature virus, slowing the spread of the virus. Type I interferons also stimulate various immune cells involved in viral clearance to more aggressively attack virus-infected cells. Type II interferon, interferon gamma, is an important activator of immune cells, figure 17. 11. A cell with viruses inside it releases signals labeled interferons. The interferons travel to 3 different cells. The interferon signals neighboring uninfected cells to destroy RNA and reduce protein synthesis. The interferon signals neighboring infected cells to undergo apoptosis. The interferon also activates immune cells. Figure 17. 11 interferons are cytokines released by a cell infected with a virus. Interferon alpha and interferon beta signal uninfected neighboring cells to inhibit mRNA synthesis, destroy RNA, and reduce protein synthesis. Top arrow. Interferon alpha and interferon beta also promote apoptosis in cells infected with the virus. Middle arrow. Interferon gamma alerts neighboring immune cells to an attack. Bottom arrow. Although interferons do not cure the cell releasing them or other infected cells, which will soon die, their release may prevent additional cells from becoming infected, thus stemming the infection. Inflammation-eliciting mediators. Many of the chemical mediators discussed in this section contribute in some way to inflammation and fever, which are nonspecific immune responses discussed in more detail in Inflammation and Fever. Cytokines stimulate the production of acute phase proteins such as C-reactive protein and mannose-binding lectin in the liver. These acute phase proteins act as opsonins, activating complement cascades through the lectin pathway. Some cytokines also bind mast cells and basophils, inducing them to release histamine, a proinflammatory compound. Histamine receptors are found on a variety of cells and mediate proinflammatory events, such as bronchoconstriction, tightening of the airways, and smooth muscle contraction. In addition to histamine, mast cells may release other chemical mediators, such as leukotrienes. Leukotrienes are lipid-based proinflammatory mediators that are produced from the metabolism of arachidonic acid in the cell membrane of leukocytes and tissue cells. Compared with the proinflammatory effects of histamine, those of leukotrienes are more potent and longer-lasting. Together, these chemical mediators can induce coughing, vomiting, and diarrhea, which serve to expel pathogens from the body. Certain cytokines also stimulate the production of prostaglandins, chemical mediators that promote the inflammatory effects of kinins and histamines. Prostaglandins can also help to set the body temperature higher, leading to fever, which promotes the activities of white blood cells and slightly inhibits the growth of pathogenic microbes. See inflammation and fever. Another inflammatory mediator, bradykinin, contributes to edema, which occurs when fluids and leukocytes leak out of the bloodstream and into tissues. It binds to receptors on cells in the capillary walls, causing the capillaries to dilate and become more permeable to fluids. Check your understanding. What do the three complement activation pathways have in common? Explain autocrine, paracrine, and endocrine signals. Name 2 important inflammation-eliciting mediators. Clinical focus, Part 2, to relieve the constriction of her airways. Angela is immediately treated with antihistamines and administered corticosteroids through an inhaler, and then monitored for a period of time. Though her condition does not worsen, the drugs do not seem to be alleviating her condition. She is admitted to the hospital for further observation, testing, and treatment. Following admission, a clinician conducts allergy testing to try to determine if something in her environment might be triggering an allergic inflammatory response. A doctor orders blood analysis to check for levels of particular cytokines. A sputum sample is also taken and sent to the lab for microbial staining, culturing, and identification of pathogens that could be causing an infection. Which aspects of the innate immune system could be contributing to Angela's airway constriction? Why was Angela treated with antihistamines? Why would the doctor be interested in levels of cytokines in Angela's blood? Jump to the next clinical focus box. Go back to the previous clinical focus box. Table 17. 5 provides a summary of the chemical defenses discussed in this section. Chemical defenses of nonspecific innate immunity. Defense examples function. Chemicals and enzymes in body fluids. Sebum from sebaceous glands provides oil barrier protecting hair follicle pores from pathogens. Oleic acid from sebum and skin microbiota lowers pH to inhibit pathogens. Lysozyme in secretions kills bacteria by attacking cell wall. Acid in stomach, urine, and vagina inhibits or kills bacteria. Digestive enzymes and bile kill bacteria. Lactoferrin and transferrin bind and sequester iron, inhibiting bacterial growth. Surfactant in lungs kills bacteria. Antimicrobial peptides. Defensins, bacteriocins, dermcidin, cathelicidin, histidins, kill bacteria by attacking membranes or interfering with cell functions. Plasma protein mediators. Acute phase proteins, C-reactive protein, serum amyloid A, ferritin, fibrinogen, transferrin, and mannose binding lectin. Inhibit the growth of bacteria and assist in the trapping and killing of bacteria. Complements C3b and C4b opsonization of pathogens to aid phagocytosis. Complement C5a chemoattractant for phagocytes. Complements C3a and C5a proinflammatory anaphylatoxins. Cytokines. Interleukins stimulate and modulate most functions of immune system. Chemokines recruit white blood cells to infected area. Interferons alert cells to viral infection, induce apoptosis of virus-infected cells, induce antiviral defenses in infected and nearby uninfected cells, stimulate immune cells to attack virus-infected cells. Inflammation-eliciting mediators. Histamine promotes vasodilation, bronchoconstriction, smooth muscle contraction, increased secretion and mucus production. Leukotrienes promote inflammation, stronger and longer lasting than histamine. Prostaglandins promote inflammation and fever. Bradykinin increases vasodilation and vascular permeability, leading to edema. Table 17. 5 learning objectives. By the end of this section, you will be able to identify and describe the components of blood, explain the process by which the formed elements of blood are formed. Hematopoiesis. Describe the characteristics of formed elements found in peripheral blood. as well as their respective functions within the innate immune system. In the previous section, we discussed some of the chemical mediators found in plasma, the fluid portion of blood. The non-fluid portion of blood consists of various types of formed elements, so called because they are all formed from the same stem cells found in bone marrow. The three major categories of formed elements are red blood cells, RBCs, also called erythrocytes, platelets, also called thrombocytes, and white blood cells, WBCs, also called leukocytes. Red blood cells are primarily responsible for carrying oxygen to tissues. Platelets are cellular fragments that participate in blood clot formation and tissue repair. Several different types of WBCs participate in various nonspecific mechanisms of innate and adaptive immunity. In this section, we will focus primarily on the innate mechanisms of various types of WBCs. Hematopoiesis. All of the formed elements of blood are derived from multipotent hematopoietic stem cells, HSCs, in the bone marrow. As the HSCs make copies of themselves in the bone marrow, individual cells receive different cues from the body that control how they develop and mature. As a result, the HSCs differentiate into different types of blood cells that, once mature, circulate in peripheral blood. This process of differentiation, called hematopoiesis, is shown in more detail in Figure 17. 12. In terms of sheer numbers, the vast majority of HSCs become erythrocytes. Much smaller numbers become leukocytes and platelets. Leukocytes can be further subdivided into granulocytes, which are characterized by numerous granules visible in the cytoplasm, and agranulocytes, which lack granules. Figure 17. 13 provides an overview of the various types of formed elements, including their relative numbers, primary function, and lifespans. A flowchart showing progression of development for formed elements of blood. At the top is a multipotent hematopoietic stem cell, hemocytoblast. This cell divides, and after division, some of the new cells remain stem cells. Others go down one of two paths depending on the chemical signals received. One path begins with lymphoid stem cells, which can either become natural killer cells, large granular lymphocytes, or small lymphocytes. The natural killer cell is a medium-large purple cell. Small lymphocytes can either become T lymphocytes or B lymphocytes. The T and B lymphocytes are medium-sized cells with a large nucleus. B lymphocytes become plasma cells, which are medium-sized cells with a large nucleus. The other option for the stem cell is to become a myeloid stem cell. Myeloid stem cells follow one of four paths. One path leads to megakaryocyte, which leads to platelets. Platelets are small flecks. The second path leads to erythrocyte. Erythrocytes are small donut-shaped red cells. The third path leads to mast cells. The fourth path leads to basophil, neutrophil, eosinophil, or monocyte. Basophils are medium cells with many dark purple spots. Neutrophils are medium pink cells with a multi-lobed nucleus. Eosinophils are medium-sized cells with many pink spots. Monocytes lead to macrophages or dendritic cells. Macrophages are large, irregularly shaped cells. Dendritic cells have longer tendons branching off of them. Figure 17. 12. All the formed elements of the blood arise by differentiation of hematopoietic stem cells in the bone marrow. A table of the formed elements. Top row reads: Formed element, major subtypes, numbers present per microliter and mean, appearance in standard blood smear, summary of functions and comments. The first row is for erythrocytes, red blood cells. There are 5. 2 million per microliter of blood, ranging from 446 million. These cells are flattened biconcave discs with no nucleus and a pale red color. Their function is to transport oxygen and some carbon dioxide between tissue and lungs. Their lifespan is approximately 120 days. The set of rows is classified under leukocytes, white blood cells. Leukocytes as a group number 7,000 per microliter of blood, ranging from 5,000. Leukocytes have an obvious dark staining nucleus and function in body defenses. They exit capillaries and move into tissues. Their lifespan is usually a few hours or days. Leukocytes are divided into two groups. The first is granulocytes, including neutrophils, eosinophils, and basophils. The second is agranulocytes, including lymphocytes and monocytes. Granulocytes number 4,300 per microliter of blood, range of 1899-50. Granulocytes have abundant granules in the cytoplasm, and the nucleus is normally lobed. Granulocytes function in nonspecific, innate, resistance to disease, and are classified according to membrane-bound granules in the cytoplasm. Neutrophils make up 50–70% of the total leukocytes, and number 4150 per microliter of blood, range 1870–300. Neutrophils have a nucleus with lobes that increase with age and pale lilac granules. They are phagocytic and particularly effective against bacteria. They release toxic chemicals from granules. Neutrophils are the most common leukocyte with a lifespan of minutes to days. Eosinophils make up 1–3% of total leukocytes. They number 165 per microliter of blood, range of 0–700. Eosinophils have a nucleus that is generally two-lobed and bright red-orange granules. They are phagocytic cells and particularly effective with antigen-antibody complexes. Eosinophils release antihistamines and increase allergies. They also help fight parasitic infections. Eosinophils have a lifespan of minutes to days. Basophils make up less than 1% of total leukocytes. They number 44 per microliter of blood, range 0–150. Basophils have a nucleus that is generally two-lobed but difficult to see due to the presence of heavy, dense, dark purple granules. Basophils promote inflammation and are the least common leukocyte. Their lifespan is unknown. Agranulocytes, including lymphocytes and monocytes, number 2640 per microliter of blood, range 1700–4900. Agranulocytes lack abundant granules in the cytoplasm and have a simple-shaped nucleus that may be indented. They function in body defenses and are grouped into two major cell types from different lineages. Lymphocytes make up 20–40% of total leukocytes and number 2185 per microliter of blood, range 1500–4000. Lymphocytes are spherical cells with a single, often large, nucleus occupying much of the cell's volume. They stain purple and are seen in large natural killer cells and small B and T cells variants. Lymphocytes are primarily involved in specific adaptive immunity. T cells directly attack other cells, cellular immunity. Natural killer cells are similar to T cells but nonspecific. Lymphocytes originate in bone marrow, but secondary production occurs in lymphatic tissue. Several distinct subtypes. Memory cells form after exposure to a pathogen and rapidly increase responses to subsequent exposure. Lifespan of many years. Monocytes make up 1–6% of total leukocytes. They number 455 per microliter of blood, range 209–50. Monocytes are large leukocytes with an indented or horseshoe-shaped nucleus. They are very effective phagocytic cells engulfing pathogens or worn-out cells, and also serve as antigen-presenting cells, APCs, or other components of the immune system. Monocytes are produced in red bone marrow and are referred to as macrophages and dendritic cells after leaving circulation. The last row of the table is for platelets. These number 350,000 per microliter of blood, range 150,005–00000. Platelets are cellular fragments surrounded by a plasma membrane and containing granules. They stain purple. The function of platelets is hemostasis plus releasing growth factors for repair and healing of tissues. They are formed from megakaryocytes that remain in the red bone marrow and shed platelets into circulation. Figure 17. 13 formed elements of blood include erythrocytes, red blood cells, leukocytes, white blood cells, and platelets. Granulocytes. The various types of granulocytes can be distinguished from one another in a blood smear by the appearance of their nuclei and the contents of their granules, which confer different traits, functions, and staining properties. The neutrophils, also called polymorphonuclear neutrophils, PMNs, have a nucleus with 3 to 5 lobes and small, numerous, lilac-colored granules. Each lobe of the nucleus is connected by a thin strand of material to the other lobes. The eosinophils have fewer lobes in the nucleus, typically 2–3, and larger granules that stain reddish-orange. The basophils have a two-lobed nucleus and large granules that stain dark blue or purple. Figure 17. 14. Neutrophils have a multi-lobed nucleus. Eosinophils have a two-lobed nucleus and distinct pink spots when stained. Basophils have a two-lobed nucleus and distinct purple spots when stained. Each type of granulocyte is illustrated with a micrograph above it. Figure 17. 14 granulocytes can be distinguished by the number of lobes in their nuclei and the staining properties of their granules. Credit Neutrophil Micrograph. Modification of work by Ed Uthman. Neutrophils, PMNs, are frequently involved in the elimination and destruction of extracellular bacteria. They are capable of migrating through the walls of blood vessels to areas of bacterial infection and tissue damage, where they seek out and kill infectious bacteria. PMN granules contain a variety of defensins and hydrolytic enzymes that help them destroy bacteria through phagocytosis, described in more detail in pathogen recognition and phagocytosis. In addition, when many neutrophils are brought into an infected area, they can be stimulated to release toxic molecules into the surrounding tissue to better clear infectious agents. This is called degranulation. Another mechanism used by neutrophils is neutrophil extracellular traps, nets, which are extruded meshes of chromatin that are closely associated with antimicrobial granule proteins and components. Chromatin is DNA with associated proteins, usually histone proteins, around which DNA wraps for organization and packing within a cell. By creating and releasing a mesh or lattice-like structure of chromatin that is coupled with antimicrobial proteins, the neutrophils can mount a highly concentrated and efficient attack against nearby pathogens. Proteins frequently associated with nets include lactoferrin, gelatinase, cathepsin G, and myeloperoxidase. Each has a different means of promoting antimicrobial activity, helping neutrophils eliminate pathogens. The toxic proteins in nets may kill some of the body's own cells, along with invading pathogens. However, this collateral damage can be repaired after the danger of the infection has been eliminated. As neutrophils fight an infection, a visible accumulation of leukocytes, cellular debris, and bacteria at the site of infection can be observed. This buildup is what we call pus, also known as purulent or suppurative discharge or drainage. The presence of pus is a sign that the immune defenses have been activated against an infection. Historically, some physicians believed that inducing pus formation could actually promote the healing of wounds. The practice of promoting laudable pus by, for instance, wrapping a wound in greasy wool soaked in wine dates back to the ancient physician Galen in the 2nd century AD and was practiced in variant forms until the 17th century, though it was not universally accepted. Today, this method is no longer practiced because we now know that it is not effective. Although a small amount of pus formation can indicate a strong immune response, artificially inducing pus formation does not promote recovery. Eosinophils are granulocytes that protect against protozoa and helminths. They also play a role in allergic reactions. The granules of eosinophils, which readily absorb the acidic reddish dye eosin, contain histamine, degradative enzymes, and a compound known as major basic protein, MBP. Figure 17. 14. MBP binds to the surface carbohydrates of parasites, and this binding is associated with disruption of the cell membrane and membrane permeability. Basophils have cytoplasmic granules of varied size and are named for their granules' ability to absorb the basic dimethylene blue, figure 17. 14. Their stimulation and degranulation can result from multiple triggering events. Activated complement fragments C3a and C5a, produced in the activation cascades of complement proteins, act as anaphylatoxins by inducing degranulation of basophils and inflammatory responses. This cell type is important in allergic reactions and other responses that involve inflammation. One of the most abundant components of basophil granules is histamine, which is released along with other chemical factors when the basophil is stimulated. These chemicals can be chemotactic and can help to open the gaps between cells in the blood vessels. Other mechanisms for basophil triggering require the assistance of antibodies, as discussed in B lymphocytes and humoral immunity. Mast cells hematopoiesis also gives rise to mast cells, which appear to be derived from the same common myeloid progenitor cell as neutrophils, eosinophils, and basophils. Functionally, mast cells are very similar to basophils, containing many of the same components in their granules, e.g., histamine, and playing a similar role in allergic responses and other inflammatory reactions. However, unlike basophils, mast cells leave the circulating blood and are most frequently found residing in tissues. They are often associated with blood vessels and nerves, or found close to surfaces that interface with the external environment, such as the skin and mucous membranes in various regions of the body. Figure 17. 15. A. Mast cells in blood. Mast cells are large purple cells, red blood cells are small pink cells with a clear center. B. Mast cell outside of blood. Figure 17. 15. Mast cells function similarly to basophils by inducing and promoting inflammatory responses. A. This figure shows mast cells in blood. In a blood smear, they are difficult to differentiate from basophils. B. Unlike basophils, mast cells migrate from the blood into various tissues. Credit right. Modification of work by Greenland Jr., Sue X, Saya DM, Liu FC, Jones KD, Looney Mr., Koi GH. Check your understanding. Describe the granules and nuclei of neutrophils, eosinophils, basophils, and mast cells. Name 3 antimicrobial mechanisms of neutrophils. Clinical focus. Part 3. Angela's tests come back negative for all common allergens, and her sputum samples contain no abnormal presence of pathogenic microbes or elevated levels of members of the normal respiratory microbiota. She does, however, have elevated levels of inflammatory cytokines in her blood. The swelling of her airway has still not responded to treatment with antihistamines or corticosteroids. Additional blood work shows that Angela has a mildly elevated white blood cell count but normal antibody levels. Also, she has a lower than normal level of the complement protein C4. What does this new information reveal about the cause of Angela's constricted airways? What are some possible conditions that could lead to low levels of complement proteins? Jump to the next clinical focus box. Go back to the previous clinical focus box. Agranulocytes, as their name suggests, agranulocytes lack visible granules in the cytoplasm. Agranulocytes can be categorized as lymphocytes or monocytes. Figure 17. 13. Among the lymphocytes are natural killer cells, which play an important role in nonspecific innate immune defenses. Lymphocytes also include the B cells and T cells, which are discussed in the next chapter because they are central players in the specific adaptive immune defenses. The monocytes differentiate into macrophages and dendritic cells, which are collectively referred to as the mononuclear phagocyte system. Natural killer cells, most lymphocytes are primarily involved in the specific adaptive immune response, and thus will be discussed in the following chapter. An exception is the natural killer cells, NK cells. These mononuclear lymphocytes use nonspecific mechanisms to recognize and destroy cells that are abnormal in some way. Cancer cells and cells infected with viruses are 2 examples of cellular abnormalities that are targeted by NK cells. Recognition of such cells involves a complex process of identifying inhibitory and activating molecular markers on the surface of the target cell. Molecular markers that make up the major histocompatibility complex, MHC, are expressed by healthy cells as an indication of self. This will be covered in more detail in next chapter. NK cells are able to recognize normal MHC markers on the surface of healthy cells, and these MHC markers serve as an inhibitory signal, preventing NK cell activation. However, cancer cells and virus-infected cells actively diminish or eliminate expression of MHC markers on their surface. When these MHC markers are diminished or absent, the NK cell interprets this as an abnormality and a cell in distress. This is one part of the NK cell activation process, figure 17. 16. NK cells are also activated by binding to activating molecular molecules on the target cell. These activating molecular molecules include altered self or non-self molecules. When an NK cell recognizes a decrease in inhibitory normal MHC molecules and an increase in activating molecules on the surface of a cell, the NK cell will be activated to eliminate the cell in distress. NK cells have both inhibitory and activating receptors. Normal cells have signals on their MHC molecules that bind to the inhibitory receptors, so the NK cell does not kill them. Cells that are infected with virus have ligands that bind to the activating receptor, This causes the NK cell to kill them. Figure 17. 16 natural killer NK cells are inhibited by the presence of the major histocompatibility cell, MHC, receptor on healthy cells. Cancer cells and virus-infected cells have reduced expression of MHC and increased expression of activating molecules. When an NK cell recognizes decreased MHC and increased activating molecules, it will kill the abnormal cell. Once a cell has been recognized as a target, the NK cell can use several different mechanisms to kill its target. For example, it may express cytotoxic membrane proteins and cytokines that stimulate the target cell to undergo apoptosis or controlled cell suicide. NK cells may also use perforin-mediated cytotoxicity to induce apoptosis in target cells. This mechanism relies on two toxins released from granules in the cytoplasm of the NK cell. Perforin, a protein that creates pores in the target cell, and granzymes, proteases that enter through the pores into the target cell's cytoplasm, where they trigger a cascade of protein activation that leads to apoptosis. The NK cell binds to the abnormal target cell, releases its destructive payload, and detaches from the target cell. While the target cell undergoes apoptosis, the NK cell synthesizes more perforin and proteases to use on its next target. NK cells contain these toxic compounds in granules in their cytoplasm. When stained, the granules are azurophilic and can be visualized under a light microscope. Figure 1717. Even though they have granules, NK cells are not considered granulocytes because their granules are far less numerous than those found in true granulocytes. Furthermore, NK cells have a different lineage than granulocytes, arising from lymphoid rather than myeloid stem cells. Figure 17. 12. Many red blood cells with a single larger cell. The larger cell is pink with a purple region that fills nearly the entire cell. The purple region is labeled perforin-containing granules. Figure 17. 17 natural killer cell with perforin-containing granules. Credit: Modification of work by Ralsted B. Monocytes, the largest of the white blood cells. Monocytes have a nucleus that lacks lobes, and they also lack granules in the cytoplasm. Figure 17. 18. Nevertheless, they are effective phagocytes, engulfing pathogens and apoptotic cells to help fight infection. When monocytes leave the bloodstream and enter a specific body tissue, they differentiate into tissue-specific phagocytes called macrophages and dendritic cells. They are particularly important residents of lymphoid tissue, as well as non-lymphoid sites and organs. Macrophages and dendritic cells can reside in body tissues for significant lengths of time. Macrophages in specific body tissues develop characteristics suited to the particular tissue. Not only do they provide immune protection for the tissue in which they reside, but they also support normal function of their neighboring tissue cells through the production of cytokines. Macrophages are given tissue-specific names, and a few examples of tissue-specific macrophages are listed in Table 17. 6. Dendritic cells are important sentinels residing in the skin and mucous membranes, which are portals of entry for many pathogens. Monocytes, macrophages, and dendritic cells are all highly phagocytic and important promoters of the immune response through their production and release of cytokines. These cells provide an essential bridge between innate and adaptive immune responses, as discussed in the next section, as well as the next chapter. Monocytes are large cells with a large purple nucleus. There is a cluster of them in a field of smaller red blood cells. A PMN is also visible with a dark, multi-lobed nucleus. Macrophages are large cells with a defined nucleus. Figure 17. 18 monocytes are large, agranular white blood cells with a nucleus that lacks lobes. When monocytes leave the bloodstream, they differentiate and become macrophages with tissue-specific properties. Credit left. Modification of work by Armed Forces Institute of Pathology. Credit right. Modification of work by Centers for Disease Control and Prevention macrophages, found in various body tissues, tissue macrophage brain and central nervous system microglial cells, liver Kupffer cells, lungs alveolar macrophages, dust cells, peritoneal cavity peritoneal macrophages. Table 17. 6. Check your understanding. Describe the signals that activate natural killer cells. What is the difference between monocytes and macrophages? Learning objectives. By the end of this section, you will be able to explain how leukocytes migrate from peripheral blood into infected tissues, explain the mechanisms by which leukocytes recognize pathogens, explain the process of phagocytosis, and the mechanisms by which phagocytes destroy and degrade pathogens. Several of the cell types discussed in the previous section can be described as phagocytes, cells whose main function is to seek, ingest, and kill pathogens. This process, called phagocytosis, was first observed in starfish in the 1880s by Nobel Prize-winning zoologist Ilya Mechnikov, 1845–1916, who made the connection to white blood cells, WBCs, in humans and other animals. At the time, Pasteur and other scientists believed that WBCs were spreading pathogens rather than killing them, which is true for some diseases, such as tuberculosis. But in most cases, phagocytes provide a strong, swift, and effective defense against a broad range of microbes, making them a critical component of innate nonspecific immunity. This section will focus on the mechanisms by which phagocytes are able to seek, recognize, and destroy pathogens. Extravasation, diapedesis, of leukocytes. Some phagocytes are leukocytes, WBCs, that normally circulate in the bloodstream. To reach pathogens located in infected tissue, leukocytes must pass through the walls of small capillary blood vessels within tissues. This process, called extravasation or diapedesis, is initiated by complement factor C5a, as well as cytokines released into the immediate vicinity by resident macrophages and tissue cells responding to the presence of the infectious agent, figure 17. 19. Similar to C5a, many of these cytokines are pro-inflammatory and chemotactic, and they bind to cells of small capillary blood vessels, initiating a response in the endothelial cells lining the inside of the blood vessel walls. This response involves the upregulation and expression of various cellular adhesion molecules and receptors. Leukocytes passing through will stick slightly to the adhesion molecules, slowing down and rolling along the blood vessel walls near the infected area. When they reach a cellular junction, they will bind to even more of these adhesion molecules, flattening out and squeezing through the cellular junction in a process known as transendothelial migration. This mechanism of rolling adhesion allows leukocytes to exit the bloodstream and enter the infected areas, where they can begin phagocytosing the invading pathogens. Note that extravasation does not occur in arteries or veins. These blood vessels are surrounded by thicker, multi-layer protective walls, in contrast to the thin single-cell layer walls of capillaries. Furthermore, the blood flow in arteries is too turbulent to allow for rolling adhesion. Also, some leukocytes tend to respond to an infection more quickly than others. The first to arrive typically are neutrophils, often within hours of a bacterial infection. By contrast, monocytes may take several days to leave the bloodstream and differentiate into macrophages. A diagram with 3 steps. The first step states, leukocytes in the blood respond to chemical attractants released by pathogens and chemical signals from nearby injured cells. An injury to the surface of the skin is labeled. Injured infected cells secrete chemical signals into the blood. Pathogens are present in the wound. Neutrophils and monocytes are in the bloodstream. And the outside of the vessel is labeled capillary epithelial cells. A resident macrophage engulfs the pathogens and releases pro-inflammatory chemotactic cytokines. The second step states, the leukocytes squeeze between the cells of the capillary wall as they follow the chemical signals to where they are most concentrated, positive chemotaxis. The leukocytes emigrate to the site of injury and infection. The chemical signals present include C5a and cytokines. The third panel states, within the damaged tissue, neutrophils release chemicals that break apart pathogens. Monocytes differentiate into macrophages. Neutrophils and macrophages phagocytize pathogens and cellular debris. Neutrophils release cytotoxic chemicals from granules into tissue. Figure 17. 19 damaged cells and macrophages that have ingested pathogens release cytokines that are pro-inflammatory and chemotactic for leukocytes. In addition, activation of complement at the site of infection results in production of the chemotactic and pro-inflammatory C5a. Leukocytes exit the blood vessel and follow the chemoattractant signal of cytokines, and C5a to the site of infection. Granulocytes such as neutrophils release chemicals that destroy pathogens. They are also capable of phagocytosis and intracellular killing of bacterial pathogens. Link to learning. Watch the following videos on leukocyte extravasation and leukocyte rolling to learn more. Check your understanding. Explain the role of adhesion molecules in the process of extravasation. Pathogen recognition as described in the previous section, opsonization of pathogens by antibody, complement factors C1q, C3b, and C4b. And lectins can assist phagocytic cells in recognition of pathogens and attachment to initiate phagocytosis. However, not all pathogen recognition is opsonin-dependent. Phagocytes can also recognize molecular structures that are common to many groups of pathogenic microbes. Such structures are called pathogen-associated molecular patterns, PAMPs. Common PAMPs include the following: peptidoglycan, found in bacterial cell walls; flagellin, a protein found in bacterial flagella; lipopolysaccharide LPS, from the outer membrane of gram-negative bacteria; lipopeptides, molecules expressed by most bacteria; and nucleic acids such as viral DNA or RNA. Like numerous other PAMPs, these substances are integral to the structure of broad classes of microbes. The structures that allow phagocytic cells to detect PAMPs are called pattern recognition receptors, PRRs. One group of PRRs is the toll-like receptors, TLRs, which bind to various PAMPs and communicate with the nucleus of the phagocyte to elicit a response. Many TLRs and other PRRs are located on the surface of a phagocyte, but some can also be found embedded in the membranes of interior compartments and organelles. Figure 1720. These interior PRRs can be useful for the binding and recognition of intracellular pathogens that may have gained access to the inside of the cell before phagocytosis could take place. Viral nucleic acids, for example, might encounter an interior PRR, triggering production of the antiviral cytokine interferon. In addition to providing the first step of pathogen recognition, the interaction between PAMPs and PRRs on macrophages provides an intracellular signal that activates the phagocyte, causing it to transition from a dormant state of readiness and slow proliferation to a state of hyperactivity, proliferation, production secretion of cytokines, and enhanced intracellular killing. PRRs on macrophages also respond to chemical distress signals from damaged or stressed cells. This allows macrophages to extend their responses beyond protection from infectious diseases to a broader role in the inflammatory response initiated from injuries or other diseases. A cell with 3 receptors. The lipopeptide receptor binds lipopeptides, the flagellin receptor binds flagellin, and the peptidoglycan receptor binds peptidoglycans. A 4th receptor, the nucleic acid receptor, which binds to nucleic acids, is found on the membrane of the phagosome. All 4 receptors have an arrow pointing to the nucleus, which contains DNA. An arrow pointing out reads: production and secretion of antiviral interferons and other cytokines. Figure 17. 20 phagocytic cells contain pattern recognition receptors, PRRs, capable of recognizing various pathogen-associated molecular patterns, PAMPs. These PRRs can be found on the plasma membrane or in internal phagosomes. When a PRR recognizes a PAMP, it sends a signal to the nucleus that activates genes involved in phagocytosis, cellular proliferation, production and secretion of antiviral interferons and pro-inflammatory cytokines, and enhanced intracellular killing. Check your understanding. Name 4 pathogen-associated molecular patterns, PAMPs. Describe the process of phagocyte activation. Pathogen degradation. Once pathogen recognition and attachment occurs, the pathogen is engulfed in a vesicle and brought into the internal compartment of the phagocyte in a process called phagocytosis. Figure 17. 21. PRRs can aid in phagocytosis by first binding to the pathogen's surface, but phagocytes are also capable of engulfing nearby items even if they are not bound to specific receptors. To engulf the pathogen, the phagocyte forms a pseudopod that wraps around the pathogen and then pinches it off into a membrane vesicle called a phagosome. Acidification of the phagosome, pH decreases to the range of 4-5, provides an important early antibacterial mechanism. The phagosome containing the pathogen fuses with one or more lysosomes, forming a phagolysosome. Formation of the phagolysosome enhances the acidification, which is essential for activation of pH-dependent digestive lysosomal enzymes and production of hydrogen peroxide and toxic reactive oxygen species. Lysosomal enzymes such as lysozyme, phospholipase, and proteases digest the pathogen. Other enzymes are involved in a respiratory burst. During the respiratory burst, phagocytes will increase their uptake and consumption of oxygen, but not for energy production. The increased oxygen consumption is focused on the production of superoxide anion, hydrogen peroxide, hydroxyl radicals, and other reactive oxygen species that are antibacterial. In addition to the reactive oxygen species produced by the respiratory burst, reactive nitrogen compounds with cytotoxic cell-killing potential can also form. For example, nitric oxide can react with superoxide to form peroxynitrite, a highly reactive nitrogen compound with degrading capabilities similar to those of the reactive oxygen species. Some phagocytes even contain an internal storehouse of microbicidal defensin proteins, e.g., neutrophil granules. These destructive forces can be released into the area around the cell to degrade microbes externally. Neutrophils, especially, can be quite efficient at this secondary antimicrobial mechanism. Once degradation is complete, leftover waste products are excreted from the cell in an exocytic vesicle. However, it is important to note that not all remains of the pathogen are excreted as waste. Macrophages and dendritic cells are also antigen-presenting cells involved in the specific adaptive immune response. These cells further process the remains of the degraded pathogen and present key antigens, specific pathogen proteins, on their cellular surface. This is an important step for stimulation of some adaptive immune responses, as will be discussed in more detail in the next chapter. Pseudopods of the larger cell engulf a smaller cell labeled infectious bacterium. The resulting vesicle containing the bacterium is labeled phagosome. This fuses with a lysosome which contains digestive enzymes. The resulting vesicle is labeled phagolysosome. Exocytosis removes the remaining debris. Figure 17. 21. The stages of phagocytosis include the engulfment of a pathogen, the formation of a phagosome, the digestion of the pathogenic particle in the phagolysosome, and the expulsion of undigested materials from the cell. Link to learning. Visit this link to view a phagocyte chasing and engulfing a pathogen. Check your understanding. What is the difference between a phagosome and a lysosome? Microconnections when phagocytosis fails. Although phagocytosis successfully destroys many pathogens, some are able to survive and even exploit this defense mechanism to multiply in the body and cause widespread infection. Protozoans of the genus Leishmania are one example. These obligate intracellular parasites are flagellates transmitted to humans by the bite of a sand fly. Infections cause serious and sometimes disfiguring sores and ulcers in the skin and other tissues. Figure 17. 22. Worldwide, an estimated 1-3 million people are newly infected with leishmaniasis annually. 1 salivary peptides from the sand fly activate host macrophages at the site of their bite. The classic or alternate pathway for complement activation ensues with C3b opsonization of the parasite. Leishmania cells are phagocytosed, lose their flagella, and multiply in a form known as an amastigote Leishman-Donovan body within the phagolysosome. Although many other pathogens are destroyed in the phagolysosome, survival of the Leishmania amastigotes is maintained by the presence of surface lipophosphatglycan and acid phosphatase. These substances inhibit the macrophage respiratory burst and lysosomal enzymes. The parasite then multiplies inside the cell and lyses the infected macrophage, releasing the amastigotes to infect other macrophages within the same host. Should another sand fly bite an infected person, it might ingest amastigotes and then transmit them to another individual through another bite. There are several different forms of leishmaniasis. The most common is a localized cutaneous form of the illness caused by L. tropica, which typically resolves spontaneously over time, but with some significant lymphocyte infiltration and permanent scarring. A mucocutaneous form of the disease, caused by L. Viania brasiliensis, produces lesions in the tissue of the nose and mouth and can be life-threatening. A visceral form of the illness can be caused by several of the different Leishmania species. It affects various organ systems and causes abnormal enlargement of the liver and spleen. Irregular fevers, anemia, liver dysfunction, and weight loss are all signs and symptoms of visceral leishmaniasis. If left untreated, it is typically fatal. A photo of a man with large skin lesions covering his face. B. A macrophage in a field of red blood cells. The macrophage has many smaller circles inside of it. Each of these has a distinct nucleus. Figure 17. 22. Cutaneous leishmaniasis is a disfiguring disease caused by the intracellular flagellate Leishmania tropica, transmitted by the bite of a sand fly. B. This light micrograph of a sample taken from a skin lesion shows a large cell, which is a macrophage infected with L. tropica amastigotes, arrows. The amastigotes have lost their flagella, but their nuclei are visible. Soon the amastigotes will lyse the macrophage and be engulfed by other phagocytes, spreading the infection. Credit, a modification of work by Otis Historical Archives of National Museum of Health and Medicine. Credit B. Modification of work by Centers for Disease Control and Prevention. Learning objectives. By the end of this section, you will be able to identify the signs of inflammation and fever, and explain why they occur. Explain the advantages and risks posed by inflammatory responses. The inflammatory response, or inflammation, is triggered by a cascade of chemical mediators and cellular responses that may occur when cells are damaged and stressed, or when pathogens successfully breach the physical barriers of the innate immune system. Although inflammation is typically associated with negative consequences of injury or disease, it is a necessary process insofar as it allows for recruitment of the cellular defenses needed to eliminate pathogens, remove damaged and dead cells, and initiate repair mechanisms. Excessive inflammation, however, can result in local tissue damage and, in severe cases, may even become deadly. Acute inflammation and early, if not immediate, response to tissue injury is acute inflammation. Immediately following an injury, vasoconstriction of blood vessels will occur to minimize blood loss. The amount of vasoconstriction is related to the amount of vascular injury, but it is usually brief. Vasoconstriction is followed by vasodilation and increased vascular permeability as a direct result of the release of histamine from resident mast cells. Increased blood flow and vascular permeability can dilute toxins and bacterial products at the site of injury or infection. They also contribute to the 5 observable signs associated with the inflammatory response: erythema, redness; edema, swelling; heat, pain, and altered function. Vasodilation and increased vascular permeability are also associated with an influx of phagocytes at the site of injury and/or infection. This can enhance the inflammatory response because phagocytes may release pro-inflammatory chemicals when they are activated by cellular distress signals released from damaged cells, by PAMPs, or by opsonins on the surface of pathogens. Activation of the complement system can further enhance the inflammatory response through the production of the anaphylatoxin C5a. Figure 17. 23 illustrates a typical case of acute inflammation at the site of a skin wound. A diagram of a wound in the skin that has let pathogens enter. Mast cells release histamines which signal to cells in the bloodstream. B. The cells have left the bloodstream. These phagocytes are engulfing the pathogens. Figure 17. 23a mast cells detect injury to nearby cells and release histamine, initiating an inflammatory response. B. Histamine increases blood flow to the wound site, and increased vascular permeability allows fluid, proteins, phagocytes, and other immune cells to enter infected tissue. These events result in the swelling and reddening of the injured site, and the increased blood flow to the injured site causes it to feel warm. Inflammation is also associated with pain due to these events, stimulating nerve pain receptors in the tissue. The interaction of phagocyte PRRs with cellular distress signals and PAMPs and opsonins on the surface of pathogens leads to the release of more pro-inflammatory chemicals, enhancing the inflammatory response. During the period of inflammation, the release of bradykinin causes capillaries to remain dilated, flooding tissues with fluids and leading to edema. Increasing numbers of neutrophils are recruited to the area to fight pathogens. As the fight rages on, pus forms from the accumulation of neutrophils, dead cells, tissue fluids, and lymph. Typically, after a few days, macrophages will help to clear out this pus. Eventually, tissue repair can begin in the wounded area. Chronic inflammation: when acute inflammation is unable to clear an infectious pathogen, chronic inflammation may occur. This often results in an ongoing, and sometimes futile, lower-level battle between the host organism and the pathogen. The wounded area may heal at a superficial level, but pathogens may still be present in deeper tissues, stimulating ongoing inflammation. Additionally, chronic inflammation may be involved in the progression of degenerative neurological diseases, such as Alzheimer's and Parkinson's, heart disease, and metastatic cancer. Chronic inflammation may lead to the formation of granulomas, pockets of infected tissue walled off and surrounded by WBCs. Macrophages and other phagocytes wage an unsuccessful battle to eliminate the pathogens and dead cellular materials within a granuloma. One example of a disease that produces chronic inflammation is tuberculosis, which results in the formation of granulomas in lung tissues. A tubercular granuloma is called a tubercle. Figure 17. 24. Tuberculosis will be covered in more detail in bacterial infections of the respiratory tract. Chronic inflammation is not just associated with bacterial infections. Chronic inflammation can be an important cause of tissue damage from viral infections. The extensive scarring observed with hepatitis C infections and liver cirrhosis is the result of chronic inflammation. A micrograph of a tubercle, which consists of many darkly staining cells that form a circular structure. Figure 17. 24. A tubercle is a granuloma in the lung tissue of a patient with tuberculosis. In this micrograph, white blood cells, stained purple, have walled off a pocket of tissue infected with Mycobacterium tuberculosis. Granulomas also occur in many other forms of disease. Credit: modification of work by Piotrowski W.J., Gorski P., Dudaszymanska J., Kwiatkowska S. Check your understanding. Name the 5 signs of inflammation. Is a granuloma an acute or chronic form of inflammation? Explain. Microconnections. Chronic edema. In addition to granulomas, chronic inflammation can also result in long-term edema. A condition known as lymphatic filariasis, also known as elephantiasis, provides an extreme example. Lymphatic filariasis is caused by microscopic nematodes, parasitic worms, whose larvae are transmitted between human hosts by mosquitoes. Adult worms live in the lymphatic vessels, where their presence stimulates infiltration by lymphocytes, plasma cells, eosinophils, and thrombocytes, a condition known as lymphangitis. Because of the chronic nature of the illness, granulomas, fibrosis, and blocking of the lymphatic system may eventually occur. Over time, these blockages may worsen with repeated infections over decades, leading to skin thickened with edema and fibrosis. Lymph extracellular tissue fluid may spill out of the lymphatic areas and back into tissues, causing extreme swelling. Figure 17. 25. Secondary bacterial infections commonly follow. Because it is a disease caused by a parasite, eosinophilia, a dramatic rise in the number of eosinophils in the blood, is characteristic of acute infection. However, this increase in antiparasite granulocytes is not sufficient to clear the infection in many cases. Lymphatic filariasis affects an estimated 120 million people worldwide, mostly concentrated in Africa and Asia. Improved sanitation and mosquito control can reduce transmission rates. A photo of a person with extremely swollen lower legs. Figure 17. 25. Elephantiasis, chronic edema of the legs due to filariasis. Credit: modification of work by Centers for Disease Control and Prevention. Fever. A fever is an inflammatory response that extends beyond the site of infection and affects the entire body, resulting in an overall increase in body temperature. Body temperature is normally regulated and maintained by the hypothalamus, an anatomical section of the brain that functions to maintain homeostasis in the body. However, certain bacterial or viral infections can result in the production of pyrogens, chemicals that effectively alter the thermostat setting of the hypothalamus to elevate body temperature and cause fever. Pyrogens may be exogenous or endogenous. For example, the endotoxin lipopolysaccharide LPS, produced by gram-negative bacteria, is an exogenous pyrogen that may induce the leukocytes to release endogenous pyrogens such as interleukin-1, IL-1, IL-6, interferon gamma, ifin gamma, and tumor necrosis factor, TNF. In a cascading effect, these molecules can then lead to the release of prostaglandin E2, PGE2 from other cells, resetting the hypothalamus to initiate fever, figure 17. 26. A diagram with exogenous pyrogen at the top. These activate leukocytes which in turn release IL-6. The leukocytes also produce pyrogenic cytokines, IL-1, TNF-alpha, ifin gamma, which lead to the production of IL-6. IL-6 signals the circumventricular organs of the brain to produce PGE2, which results in fever. The temperature-dependent feedback on cytokine expression decreases the production of IL-6 in a negative feedback loop. Figure 17. 26. The role of the hypothalamus in the inflammatory response. Macrophages recognize pathogens in an area and release cytokines that trigger inflammation. The cytokines also send a signal up the vagus nerve to the hypothalamus. Like other forms of inflammation, a fever enhances the innate immune defenses by stimulating leukocytes to kill pathogens. The rise in body temperature also may inhibit the growth of many pathogens, since human pathogens are mesophiles with optimum growth occurring around 35 degrees C, 95 degrees F. In addition, some studies suggest that fever may also stimulate release of iron-sequestering compounds from the liver, thereby starving out microbes that rely on iron for growth. 3. During fever, the skin may appear pale due to vasoconstriction of the blood vessels in the skin, which is mediated by the hypothalamus to divert blood flow away from extremities, minimizing the loss of heat and raising the core temperature. The hypothalamus will also stimulate shivering of muscles, another effective mechanism of generating heat and raising the core temperature. The crisis phase occurs when the fever breaks. The hypothalamus stimulates vasodilation, resulting in a return of blood flow to the skin and a subsequent release of heat from the body. The hypothalamus also stimulates sweating, which cools the skin as the sweat evaporates. Although a low-level fever may help an individual overcome an illness, in some instances, this immune response can be too strong, causing tissue and organ damage, and in severe cases, even death. The inflammatory response to bacterial superantigens is one scenario in which a life-threatening fever may develop. Superantigens are bacterial or viral proteins that can cause an excessive activation of T cells from the specific adaptive immune defense, as well as an excessive release of cytokines that overstimulates the inflammatory response. For example, Staphylococcus aureus and Streptococcus pyogenes are capable of producing superantigens that cause toxic shock syndrome and scarlet fever, respectively. Both of these conditions can be associated with very high life-threatening fevers in excess of 42 C, 108 degrees F. Check your understanding. Explain the difference between exogenous and endogenous pyrogens. How does a fever inhibit pathogens? Clinical focus. Resolution. Given her father's premature death, Angela's doctor suspects that she has hereditary angioedema, a genetic disorder that compromises the function of C1 inhibitor protein. Patients with this genetic abnormality may have occasional episodes of swelling in various parts of the body. In Angela's case, the swelling has occurred in the respiratory tract, leading to difficulty breathing. Swelling may also occur in the gastrointestinal tract, causing abdominal cramping, diarrhea, and vomiting, or in the muscles of the face or limbs. This swelling may be non-responsive to steroid treatment and is often misdiagnosed as an allergy. Because there are 3 types of hereditary angioedema, the doctor orders a more specific blood test to look for levels of C1-INH, as well as a functional assay of Angela's C1 inhibitors. The results suggest that Angela has type I hereditary angioedema, which accounts for 85% of all cases. This form of the disorder is caused by a deficiency in C1 esterase inhibitors, the proteins that normally help suppress activation of the complement system. When these proteins are deficient or non-functional, overstimulation of the system can lead to production of inflammatory anaphylatoxins, which results in swelling and fluid buildup in tissues. There is no cure for hereditary angioedema, but timely treatment with purified and concentrated C1-INH from blood donors can be effective, preventing tragic outcomes like the ones suffered by Angela's father. A number of therapeutic drugs, either currently approved or in late-stage human trials, may also be considered as options for treatment in the near future. These drugs work by inhibiting inflammatory molecules or the receptors for inflammatory molecules. Thankfully, Angela's condition was quickly diagnosed and treated. Although she may experience additional episodes in the future, her prognosis is good, and she can expect to live a relatively normal life provided she seeks treatment at the onset of symptoms. Go back to the previous clinical focus box.